Hello. So I'm here in this video to explain about more cell-to-cell interactions, since some of you guys have asked me. So let's think about cell-to-cell interaction, especially in eukaryotes. Eukaryotic organisms like plants and animals, our cells need to communicate with each other. Because imagine it's just a huge amount of small little cells that are all working together to keep your body, running. So they need to talk with each other in some sort of way. So that's why cell-to-cell interactions are very important, because it is some sort of way of communication. So the first type of cell-to-cell interactions are basically the surface proteins. And these surface proteins kind of are these identities. They give identity to cells like it kind of a passport gives identity to us or a Hawiye or a national identity card. So these are basically surface proteins. They give out identity and help basically cells identify each other. First one, for example, are like glycolipids. Glycolipids are literally lipids that have a lot of carbohydrates in them. And this really serves to basically cells to give an identity. And I'll give you here an example specifically with blood types that I've used a lot. So how people actually have, group A, or not, sorry, type A blood type. Some others have type B, or some others have type A and B, and how or O even. So how can you then distinguish them is looking at the glycolipids because type A red blood cells have this certain type of glycolipid, while the others have a different type of glycolipid, and it really helps kind of identify which blood type it is because sometimes you don't want to mix them together. So this kind of gives you a very important kind of, let's say, identification. Another one that is very important are MHC proteins. And these proteins really kind of help your normal cells interact with your immune cells. Because your immune cells, they have one big job, which is to protect you from anything foreign, any microorganism that's, coming in to do harm. So how it's able to do that, it's able to communicate with cells, and the cells will tell them like, "Hey, I am a healthy cell from the body. Please do not kill me." And they do this. They do this through MHC proteins. Well, imagine a cell that actually got hijacked by a virus. So a virus comes in and it attacks it. Now that is becoming non-self because it's hijacked. It's not, something some other organism took charge of it now. So we see that as non-self. So the immune system is gonna then talk to the cell through the MHC and be like, hey, are you okay? And the cell's gonna be like, no, I'm not okay because there's a virus hijacking me. So that's one way of cell-to-cell interaction in way how MHC proteins help immune cells and normal cells speak to each other and say if they're actually healthy or not. Another form of cell-to-cell interactions are cell connections. So these are proteins that are literally trying to kind of push cells together. So that's what they're doing. And there's three different types. There's adhesive junctions, septate or tight junctions, and communicating junctions. And we'll start with the first one, which is adhesive junctions. These are proteins like adherent junctions or desmosomes that what they do is they attach cytoskeletons of neighboring cells together. That's what they do. Attach the cytoskeletons of neighboring cells. So I'll give you here an example. You have this cell over here with this cytoskeleton, specifically your microtubule, and this other one with your actin. How do you connect them? Is when you have one cadherin from basically this one cell that's attached to the actin, and it attaches to the other cadherin from the neighboring cell, which is also attached to the microtubules. So the microtubules are in some way indirectly attached through these cadherins. So whatever happens here, the actin filaments can actually sense it, and they can actually, for example, if this is contracting, this could contract also. So it serves in some sort of way, some communication between the uh, the basically cytoskeleton of cells, or also basically into the extracellular matrix. So this extracellular matrix is the matrix that's outside of the cell. All these basically glycoproteins that give the cell support. So those are adhesive junctions. Now you have more septate or tight junctions. These guys, very similar to adhesive junctions, what they're trying to do is push cells together. But these, as their name is, it's tighter. So here, what you're trying to do is you're really trying to push these cells way much more closer with each other. That's what you're trying to do. You really don't want to have any space. So where would you see this? Well, you would see this actually, for example, in your esophagus. So the esophagus is literally this really nice kind of structure that we have that connects to basically our stomach and our mouth, and where basically our food passes to go to the stomach. So that is actually one cell layer thin. So very thin, and it's one. It has basically one layer of one cell. It is super crucial that there's no single opening there. Because imagine if the esophagus had openings, well, food will actually pass through those openings and basically go somewhere else. You don't want that. You want them to go straight to your stomach. So how do you do that? Is you have these tight junctions, these proteins here, tight junctions, which they wrap around in some sort of way, just like kind of a bracelet around these cells, and they push them together. They push them so tightly together that there's basically no space between them. And this is very important. So look, these are the tight junctions. Here, these are the adhesive junctions. These are the ones we just talked about, like the cadherins, that are literally connecting the um, cytoskeleton of one cell to the other. So that's basically, these are not as tight as this. This is literally proteins are going around and they're really pushing the cells to go super tightly and have no actually openings. These are just connecting the cytoskeleton to the other cytoskeleton or the extracellular matrix. They're just trying to connect them, not to push them so close to each other. Here they are. And then you have the final one, which is in some way trying to connect actually two cells together via actually a channel. So this is a very different type of basically connection, because here you're just pushing them together to to forcing them together. Here you're basically making the cells their cytoskeleton communicate with each other. Here no, here you want actually things to pass through, because now that you open this channel, you're letting basically ions, macromolecules go through. So these cells can communicate with each other through ions, through macromolecules, because you have an opening. And in this case here, you have these connexons, which are literally these proteins that create these kind of tubes or channels between two cells. So you have tight junctions, which are proteins that really push two cells super together. And think about that as your esophagus, this thing in in your throat that's helping pass food through. You don't want any food to go through it, so you're gonna push the cells super together so that you create a tight junction. So you're trying to communicate with the cells and push them together to make zero spaces. The second is adhesive junctions, and these are trying to make your cytoskeletons of two different cells communicate with each other. And this could be very important, for example, with motility. If basically one cell is trying to move and telling the other cell to move, the cytoskeleton is basically that's gonna make the whole cell move. So if one cell moves, the other cell through the cytoskeleton will perceive it. Because of course of these cadherins that are connected to each other through the cytoskeletons, they'll be able to communicate that. And then you have these communicating junctions, which are literally channels that are open and allow certain ions and macromolecules to pass through. So these cells are communicating with each other through macromolecules and ions on all other molecules. In plants, you basically have something very similar to these communicating junctions. So in basically animals, you have these connexons, while in plants you have something called plasmodesmata. And plasmodesmata is literally almost the same as this, but it's between two plant cells. It's literally when you have, for example, here in this case, you have the smoothie R that goes through one cell to the other and creates this channel where things can pass through one cell and the other. So they're literally the similar gap junctions, but they're just the plant version of it. So they're connecting the two cytoplasms from this to this. Well, same thing goes here. They're connecting the cytoplasm from this cell to this cell. Same thing. So it's just more the plant version.